we could do genetics in Burmese. And so I had actually gone to Europe and found some pedigrees there, talked with some people there. Does anybody know Beatrice Wood? Met her and stuff. Went to Belgium and was handed craniofacial defect kittens in formalin. And it became quite clear that to have the defect that took 2 copies, it was a recessive trait. But if you had 1 copy, you certainly probably had a short face, but could you quantify that in some way? I went to Johns Hopkins, talked to a very prominent geneticist there called Victor McCusick, showed him these kittens, does this match anything in humans? And he, no at the time, but we thought, could we do measurements? Could we do x-rays? So we were trying to focus on, can we figure out who the carriers are? And in the end, we figured out, in the end, there was no easy way to do it, right? So that's where things kind of stayed, because we had no pedigrees. We just had a few affected kittens at the time. And so then people like Karen and Joanne Arnett and a few other people, there was a call to collect samples. And so remember, this was 5 to 10 mils of blood. Yeah, there was the call for 5 to 10 mils, because we didn't have buccal swabs invented at that point, right? So 5 to 10 mils of blood, and some of the first samples, you know, some of the people are gone now, I'm sorry to say, but we collected up cats from different breeds, Tonkinese, American Shorthairs, Bombays, as a background baseline. So by the time I left the NCI, we maybe had 1,600 samples from various different projects. We were building up the pedigrees at the time so we could do the old style linkage analysis. The microsatellite markers were just being developed in the laboratory. So by the time I left the NCI, we maybe had 300 of those microsatellite markers, and we had a very good craniofacial defect pedigree. And so that took 5 to 7 years to collect and to develop those markers. And then once I moved to Davis in '99, I asked that I would take this project with me. And so when I got to Davis, we were able to actually do the linkage analysis, and that's when we localized the gene to a specific chromosome. And so that was with Carolyn Erdman. And so we localized it to a specific chromosome, but then it sat for a while. Carolyn left the lab. We didn't have the right people to keep working on it and stuff. And so, and then the DNA arrays came along. So it's been a long, arduous process, but they've always kind of been there. But it's also been hard because there's never always been one focused person working on it at the same time as well. And that's because of consistency in funding. But at one point, then we got a lot of funding help, and that moved it, jumped it quite a long bit farther. But the DNA chips here in the end are and somebody like Barbara, who is clever enough to really diligently look at and analyze the data. Pardon me? All of our prayers. All your prayers, yeah, all your prayers. People who have consistently sent in samples. I know there's a lot more, you know, there's some people that hung with you. I know there was a lot more kittens being produced that ever got sent into the lab and stuff, so, but.
Les, you may just want to take a minute and tell them how that DNA chip developed, because there's other, you know, groups of people who contributed to that.
Right, yeah, so I mentioned it a little bit at the symposium. So the DNA chip, the NIH first did some sequencing of the cats, and that has helped quite a bit, but it was a very minor sequencing effort. It was just very rudimentary. And then along the way, Hill's Pet Food came along. So Hills and Purina and these guys are interested in figuring out the genetic traits that are involved with nutrition. They would love to figure out why cats get fat. They would love to figure out why cats are diabetic. They would love to figure out why a cat likes this food over that food, right? And once they find that out, they're not going to tell anybody, and they're going to sell their food. That's what they want to figure out, right? So they started doing some sequencing of the cat as well. And then Hill's Pet Food said, all right, we're going to make 1 million. Now, why they did this, I don't know. Maybe because they couldn't do it internally. They said, here's $1 million. Cat community, do what you want with it. So Morris Animal Foundation put together a committee, and let me tell you, if you think you guys fight, you had to seen this committee fighting, all right? So you guys are nothing compared to the fights I had on this committee. And now the committee said, absolutely, we want a DNA chip, right? So that was easy, but then we had to fight which cats are we going to sequence so we can find these SNPs, and we used all this breed stuff, and that all helped with that and everything. And in the end, we decided we're going to have a chip. So that $1 million was enough money to produce 4,600 DNA chips. That chip looks like a microscope slide, all right? But it's made out of metal. But you can put little DNA, pieces of DNA on it, and you put 63,000 in one teeny little, like the size of the head of an eraser, all right? And those are then spotted onto this array. And so you can test 12 cats at a time with 63,000 genetic markers all at once. And you get the data back in 3 weeks. Okay, I could never accomplish this. Remember, I'm fighting to do 300 markers on a pedigree of 200 cats, and it's taken me my lifetime to do this. And now you can do it in a week. It sucks, you know, because... But now the data analysis is important now, you know, so now you have this overwhelming amount of data, so now you got to find students that can run computers instead of pipettes, you know. Now we get our undergraduates, such as the young Kelsey Hickey back there, right? Woo! So we bring her out from Pittsburgh, put her in the lab, make her be a minion for a few months, you know. And she helps us isolate all these data. So the DNA isolation now is easy. The generating the data is easy. The analysis is still very tricky and stuff.
And the Cat Health Network.
Yes, and so we have this chip, but now what are we going to do? Who gets the chip? Huh? Who's going to get them, right? So then the Cat Health Network got put together. So the Cat Health Network was Morris Animal Foundation, the Wynn Feline Foundation, AVMF, American Veterinary Medical Foundation, and was AFP was also in there as well. Who really puts forward the money? You know, Morris is pretending, oh yeah, that Morris didn't put any money towards this. It's Hills, but Hills Morris is kind of a derivative of Hills actually in the end. So, but Morris is like, we're in charge, but you know, they didn't put any money forward. It was Wynn Foundation that put money forward so that, yeah, you can have a chip. Here's your chip. Now what are you going to do? Who's going to isolate the DNA? Who's going to pay Kelsey Hickey to isolate that DNA? Who's going to pay for the people that are sitting down just doing the analysis, just to tell you the location of where the gene is? Because that's all that chip does. It tells you it's on chromosome B4 in this area of the chromosome. Now you got to look under that gene, under that location, figure out what gene it is, and that's where Barbara comes into play, right? She looks under there and says, oh, there's this gene, that gene, this gene. I'm going to read. So she spends hours and hours reading background and literature until you come across the right gene, and then you go into the laboratory. The Wynn Foundation helped fund that part, and that was really the part that found the mutation and stuff as well. So in the end, Wynn Foundation puts up the most money that really pushes these array projects together. Now the $1 million from Hills, that was for me a gift from God, because the pig people hate us. The pig people hate us because they needed to get $1 million to make their chips, right? And then we talked to the company in Nebraska, and Nebraska was like, yeah, these chips, they're old. Tell you what, you allow us to run all 4,000 chips, we'll give you a price of $25 per animal, because it still now costs money to assay the chip. And it's $25 per animal. Again, that's what the Wynn Foundation money did, right? So we have the chips for free. We still have to spend money isolating DNA samples. We still need to pay someone to do that, and we still have to assay the chip, right? So that's where Wynn money comes from. And then, but then they gave us the price of $25. Well, the pig guys were doing it for $50. So when they found out we got our chips for free and at half the price to run them, you know, they were pretty annoyed. You know? So It's like, too bad for you. But so sometimes, sometimes the cats are a few steps behind in genetic research, but we get to do things sometimes a little more efficiently because they've made all the mistakes. And that's what I do. I go to genetic meetings for humans and for cattle and for horses, watch what they do, and steal it. And I say, yeah, that's what we need to do, but we won't make all those mistakes in the meantime, right? And we get a better price. So we're a little slower, but we're more efficient, actually. So, yeah. Anyhow.
What is next-gen sequencing for animals?
Next-gen sequencing, Davis wants to be the first place to sequence a cat for its health problems.
Yeah, the data.
Yeah, the data. So what the problem? So probably, I would say, most of you in this room might have your DNA sequenced as part of your healthcare plan. I know probably I will.
Everybody. All the molecular labs are doing next-gen sequencing.
So
We're trying to get it on.
they're trying to get the price for completely doing your complete DNA sequence down to $1,000, and they're nearly there. Right now it's about $5,000 to $10,000. Yeah, but
as it becomes more prevalent.
right. And so what you need is the database to compare to. So the cat doesn't have the best database yet, but they've already done sequencing for dogs. Take an individual dog, I'm going to sequence you, and then I'm going to try to find the DNA mutations that cause you to be sick, right? They've done it for cattle. Davis wants to be the first one for the cat. What the sequencing company group wants to do is they want to do a cancer project. So we might try to send off one of these mediastinal lymphomas or something like that, right? So I want that to be the future of Davis, future of the VGL. Forget all this crap. We'll just sequence your whole darn cat, And but it'll come. It comes faster than you think. In my lifetime, we went from millions and millions of dollars to sequence the human genome, and James Watson's the guy that discovered DNA. We thought he was crazy as a balloon, man. He was up there saying, we're going to sequence a genome. And you ever watch Simpsons, Mr. Burns on Simpsons? Guy that has that snicker? That's James Watson. That is him. And so you're listening at a meeting and this guy's like this, and you oh my God. But in the end, he was right. In the end, he was right. He had vision. And now, I think probably within the next 5 to 10 years, we'll be sequencing cats and dogs as part of their health care plan. And I'm hoping Davis will be one of the first to do it. It will probably come for us. I've actually, some of my other slides are a company called 23andMe. Anybody heard of these companies, 23andMe? You can send in your own DNA and get your own mutations. I use that as part of my presentation, so I have my own. And this is something you guys should know. Everybody has mutations. Everybody has something. So if you bred back with your parents, it would show up, right? Okay. So we all have that. We all have that. I didn't hear what you said.
I think the estimate has been every human being has at least 300 or more lethal mutations. Wow.
I don't think it's as high as 300, but yes, every one of us in this room have some type of lethal mutations to us, and that's called our genetic load. And so we all have it. So that means all the individual cats have it as well. So that project's called 23andMe. Why 23? Anybody know? 23? 23 chromosomes. How many chromosomes does a cat have? 19. All right, 19 chromosomes for a cat. You get a prize. All right. You guessed, didn't you? 19.